Hello and welcome to the last presentation in this room. We're going to present 12 Legs Infinite Possibilities using hexbooks to model physical systems. My name is Ashley.
I'm Sophia.
I'm Carlos.
And here are our hexbooks.
So the first thing we're going to talk about is motivation. So why are we changing these physics labs in the first place? So what are the current issues with IPLS labs? And for context, IPLS stands for Introductory Physics for Life Sciences. So with previous research from Professor Holmes, we have found that students don't learn a lot from traditional labs. The traditional labs don't teach them how to build transferable research skills, which in STEM-based courses is very important. Also, there's a huge disconnect between biology and physics. And for a class that's teaching physics to life science students, that should be a main focus point. And also, they can be a lot more fun than speaking for experience I took in class.
So there's previous research that has kind of come up with a good solution for these issues that are faced not only by our FPLS lab, but other labs around the country. And these are called course-based undergraduate research experiences, also known as CURES. And they're an extension of traditional undergrad research experiences, like the next program we're in right now, except they're more accessible to students because they are embedded inside courses in the curriculum. So they actually came out of research in biology and physics education, and the focus is on helping develop research skills instead of kind of reiterating material. And other introductory physics classes here at Cornell, such as Physics 1110 and 2210, have already successfully implemented these, and we want to kind of follow their model and bring it to this course.
Okay, so to summarize, so before the labs were very cookie cutter, they gave students like a notebook of set instructions and they had to follow them exactly. And because of this, there was like little to no room for students to exercise their own creativity and come up with their own research questions. And also there was no connection to biology at all. So now we want to design a focused lab that's based on focused biology and physics concepts instead of trying to cover the entire physics course, which is too much to cover in a lab course. We only have like 6 labs or 7 labs a semester. We want to allow students to design their own experiments with what interests them and what interests them in physics and biology. And also this will help bridge the gap between biology and physics. So the main goal of this entire project is to create an interactive lab that's relevant to both physics and biology.
So the solution are hexbugs, which are little robots that model random motion. And this is a short video of them. And so the left is a sped up video of the hexbug moving in an arena, and the right is a simulated animation of particles moving randomly in a chamber. So you might ask the question, why use hexbugs in the labs? So previous research has analyzed the motion of hexbugs and the potential educational applications of them. We are particularly inspired by the previous research done by students in Pomona who developed labs modeling the ideal gas law with hexbugs. So random particle movement that is modeled by hexbugs is common in both physics and biology, and they relate to concepts such as ideal gas law, fluid dynamics, diffusions, and etc. Not to mention that both physics and biology care about modeling chaotic systems to a degree where physicists are writing papers about hexbugs for other physicists, and other biologists are emphasizing that diffusion, which is modeled by these hexbugs, are crucial in biology education. So in summary, hexbug serves as real-world simulations of these relevant biology topics, and we believe that could help labs be more effective for the students.
So our next section in our presentation is about model development. And kind of the main question we're asking in this section is, what kind of experiments can we actually have students do with these hexbugs? So to start, we want to model active Brownian motion, which is the random movement of particles. And this is kind of the baseline basis for all the other experiments. And this has been verified before in other papers, but we want to re-verify for ourselves and make sure it is accurate. So active Brownian motion, it has a few characteristic traits. The first is that every change from the last position is independent of each other. So basically it just moves randomly around, and its displacements are actually distributed in a normal distribution. And there's also an equal tendency for the particles to move in any direction. So there's a little picture just showing a sample of what Brownian motion could look like. And it is caused by collisions of particles. And in this instance, it is actually active Brownian motion. So that means that the particles are self-propelled in addition to being driven by collisions. And that is the case for hexbugs.
And for the biological context of this, it helps model motion of bacteria in viscous environments like E. coli and ion movement within cells in the body.
So to test this, we released 15 bugs in a 60 centimeter by 60 centimeter arena for 5 minutes. And this is a short video of them moving within the arena. And the reason for the particular shape of the arena, we will discuss a little bit later. But to analyze their motion, we analyze the displacements between the time steps to check if they actually model Brownian motion. And by that, we mean their individual one-dimensional displacement distribution would model a very good normal distribution, as you can see in the graph here.
Okay, so one of the labs that the students can do is a diffusion lab. And for the biological concept of this, diffusion is just the movement of molecules from an area of high to low concentration. And the rate of diffusion is proportional to the concentration difference. So more molecules in areas of high concentration will collide with each other and move to spaces where there's low concentration. In the body, particles and molecules like water, oxygen, and carbon dioxide, for example, there's many more that I'm not going to list them all, diffuse across cell membranes along their concentration gradients. And then I have a biological application of diffusion here called osmosis. So when the concentration of water is lower outside of the blood cell than inside, the water will rush out. And then when it's the same, it will be an even exchange. And when there is a high concentration of water in the cell versus out, the water will rush out and shrink, and vice versa.
So for the experiment that concerns the concept of diffusion, we released the bugs from one side of the arena.
And our analysis of diffusion was that we tracked the number of bugs on both sides of the arena. We had a membrane that we constructed in the middle with openings, and we tracked the number on each side. And we found that it follows an exponential decay model as we expected from just equations that model this.
So here is a short video of the hex bugs. You could see this Lego membrane has 3 openings, and you can see some of them just slipping in and out.
Okay, and then an extension of diffusion onto a more complex model is diffusion with a membrane. So the permeability of a membrane is proportional to the rate of diffusion. So the more openings or ion channels that a membrane has, the faster ions can move through it. And yeah,
so for this, our experiment was to change the number of openings in the Lego membrane barrier that we had constructed.
And to analyze the data, we tracked the number of crossings that corresponds to the rate of diffusion across the membrane. And here are some of the Lego membranes with the openings, as you can see. And the data showed that as you can see in the first column, that as the number of openings doubled, you can see in the last column that the rate per minute of these crossings or that the number that corresponds to the rate of diffusion approximately doubled.
Okay, and then we're also giving students the opportunity to come up with their own models based on their own biological interests. I'm concentrating in neurobiology, so I'm interested in action potentials. And so basically, action potentials are a rapid change of voltage across the cell membrane. They will make your neurons fire, will make you help function as a normal human being. So I'm going to try to simplify this as much as possible for everyone to understand. But in the body, you have a resting membrane potential, which is negative. However, stimuluses happen with neurotransmitters, and they let a little bit of positive particles go in. And these things called voltage-gated ion channels open at certain potential differences. So the slight rise in positive potential causes them to open and causes a rush of positive particles into the cell. It reaches a maximum. These channels close, other ones open to allow them to move out of the cell for repolarization. And so I decided to come up with a very conceptual model of this, and this is just my proof of concept model. So the stimulus is going to be the students loading the bugs onto the pathway. The ramp is the ion channel, and the box here is the neuron or cell. It's going to reach a maximum positive potential or maximum number of bugs, and then it will eventually tip over and reset itself like in repolarization.
Wait.
And it's kind of a slow process, but it'll happen.
Oh, yeah, and this is a proof of concept model. We are still in the process of kind of 3D modeling this and trying to transform it into something that's
more sturdy.
Yeah.
In the next section, we will actually talk about the infrastructure, and we're going to answer the questions of what we actually use to conduct our experiments and how we adapted our methods for the students to use in future labs. So some of our end products include Hexbug Tracker, which is an interactive app we developed to track the motion of hexbugs. notebook style lab with that has easy to use code for beginner coders, considering the fact that most life sciences / biology students are not familiar with complex coding. And also just basic materials and parts that can be used to construct the models and the demo systems.
So our first kind of product we created was Textbooks Tracker, and this started initially as kind of our solution to just a limited selection of free tracking software online. In the beginning of our analysis, we tried to use a software called Tracker, which allows you to track motion of objects, and it didn't work well. It was very difficult, especially for the motion of hexbug, as it is random motion, and it kind of tries to follow a path. It was not a good solution, and we really couldn't find any better alternatives. So we started using open source packages and code to build our own kind of tracking software so we can get data for these textbooks. And this is just different files that we kind of iterated on there. So we were using that for our own purposes, but we also realized that students themselves, we can't assume really any familiarity with code or anything. So we decided to adapt this for student use. And we transformed our code and tracking software into an app for the students to use. And we gave it an interactive interface for selecting videos, so it's simple and easy for students to understand with no code needed. So as you can see, they just click on the app, it'll ask them to select a video file and processing FPS. And then there's just a window that pops up and it kind of guides the students through the process of collecting data for the textbooks.
The next is these notebook style code, as we mentioned. And this allows students with almost no coding experience to follow along with the analysis process. And as you can see, these Jupyter / Google Colab type notebooks allows you to have these normal texts, which can be used for instructions, but also these code blocks where the students can simply click run and it will spit out analysis. And it is currently used in 1110 and 2210 labs. It allows, as I said, for lab instructions and guidance, and it is packaged with pre-made functions that we used during our own analysis.
Okay, so what materials are we going to give the students to use? So our main material here is hexbugs. Each group is going to get 10 to 15 hexbugs. We found this was the best number of hexbugs to maximize the data, but also minimize the cost of buying 2,000 hexbugs for all students. And each group is going to use hexbugs from the previous group and the previous lab section, so this also minimizes the cost. We're also going to give them simple construction materials to use in case they want to build their own models that we don't have the 3D printed materials for. So Legos, cardboard, and tape, just how I built my own potential model. And then for the basic models of diffusion and random motion, we have this cloud-shaped arena. We found from previous research that this was the best shape to help redirect hexbugs towards the middle of the arena. With regular rectangular shaped arenas, the hexbugs would get caught in the sides, and then it would mess up the data collection.
We obviously want to talk about our conclusions for the last section, and we want to answer the question of what comes after this. So to reiterate our point about why we should use hexbooks in labs, hexbook serves as real-world simulations of relevant biology and physics concepts, and it can help labs be more effective. And we believe that by developing research-based labs with hexbooks to model random complex systems that are relevant for both biology and physics, we can maximize the learning and skill building of students in IPLS. We want to wrap up the slideshow with what we have learned from this research experience. And I believe I got to experience the process of iteration and record keeping for these long-term projects.
I feel like I got a deeper understanding of the physical mechanisms behind biological systems and the connection between biology and physics, which is important to me since it's my major and also the goal of the class. And I also got experience with building accurate models.
And I feel like I got a lot more rich coding experience with developing software and tools to use, and also implementing different data analyses for different systems.
We want to thank our mentors, Professor Holmes, Dr. Zimmerman, Mark, and everybody in the Cornell Physics Education Research Group. And here are our works cited, and we would love any questions.
I'm curious, this is super interesting, and I love the examples. I'm really curious kind of if you've mapped like the whole semester curriculum to where you could incorporate hexbugs. Like, because when I think of my physics, there was like mechanics, which was like ramps and blocks,
Yes.
and waves, and electromagnetic,
Yes.
but you can seemingly do some electrical
Yeah.
stuff with these.
The
And
only
you can do like some of these gas things, but we didn't even do. Like-
So these labs are special for the 2207 to 2208 or potentially.
Yeah.
And Sophia can talk about
2207 is like kinematics and everything, and then 2208 is electrics and mechanics.
Right.
So you can model the movement of positive particles with hexbugs. You can't really with negative particles because you can't have like negative space.
But I think the whole point of the lab isn't to cover
All of
every
it
concept.
is just
So this is like kind of like an additional piece where you can develop skills
Yeah.
and reiterate on some topics, but it shouldn't serve to just relearn all the topics covered in lectures. That would come in the class.
Did you do traditional mechanics?
Yes.
in some ways, yes. But the concept is more that flow is a huge part of biology and physics, and a lot of different physics concepts are connected by the notion of flow, which can be shown with textbooks. So it's not covering everything, but it's covering a lot of key points, especially points in biology and things that will be on biology graduate programs and stuff like the MCAT. It's a positive thing.
Really great presentation. So when I think of cures, I think of one of the integral parts is having students ask and answer their own research questions, analyze their own data. And I guess something I'm wondering is how do you see hexbugs as a way to support this cure model of open inquiry, or do you see it more as being kind of like a guided inquiry, like you provide students with materials, you provide students with the shape rather than having them determine the shape, and then you have them collect the data and analyze it. Which one do you see as being more?
We see it as both. So as our plan, so as we mentioned, the first few classes would be kind of an introduction to hexbugs and this like model building process. So it would be guided. And I think potentially the first two experiments would be the active random motion and the diffusion. And then after that, the rest of the course would be devoted to them trying to figure out, like, ask research questions, test the models that they built before, and just like build their own models.
We're planning on showing them the other models we also made like a piston for a gas chamber and stuff like that we didn't show in here. And we're gonna say like these are the options, things you can do or want to explore if it inspires you. Just come up with your own research question that's related to a biological concept. We'll be there if you need support.
Nice.
And then like free reign, do what you want. And just the course. And we'll check in. Yeah.
Planetary versus exploratory. How?
I think when you explore new concepts and build systems that you can change yourself, it helps you come up with the explanation. And by changing things, you can see how different things affect others, and that will help you understand the models and graphs and math better. So exploring helps you explain.
And I don't think we're not looking for correct answers from students. We're just looking for them to be able to support the data and analysis they gathered and explain why they think the way they do. It doesn't need to be correct.
How much is a hexbug?
The model on Amazon has toys for cats and kids, so $2
Yeah.
We're very talks.
Yeah.
You gotta talk about changing on the Xbox because they're actually Xbox now that they've been bought. So we'll talk about it a little fine. We're working at it.
Maybe we can get like a bulk purchase.
That's what we're doing.
Yeah.
Let us know.
Thank you.